Alright everyone, I want to talk about the digestion of, well, the absorption really, of proteins. Now let's remember that it starts, and this digestion starts in the stomach with pepsin. Continues in the small intestine with trypsin. Here I drew them broken down a little. But notice how they're not monomers yet. The brush border, the enzymes on the brush border, I should say, break it down into monomers, break the proteins down into amino acids. And then, but we still need to get the amino acids into the cell. Now, are the amino acids, are they hydrophilic or hydrophobic? They're hydrophilic. So can they just travel right through? They cannot, can they? Alright, so what happens is we establish a gradient. We use a sodium-potassium pump on the basal membrane to establish a gradient. So we have, see, sodium in, potassium out. And then, because we have, after this, more sodium inside the cell than outside, we're able to use that gradient and pump sodium out while pumping an amino acid in. So remember, down here, this is primary active transport. So here is secondary active transport. We're using that sodium gradient to pump an amino acid in. The amino acid travels through the cell and out the basal membrane, and then is able to dissolve into the blood, diffuse into the blood, because it is hydrophilic. Does that make sense? So we had that sodium-potassium pump. We use the gradient. This is called secondary active transport. Alright? It's able to diffuse into the blood because it's hydrophilic. And remember the enzymes. Pepsin in the stomach, trypsin in the small intestine, brush border enzymes, breaking it down into single amino acids that can then travel through using that sodium gradient. Does that make sense? It's very similar to carbohydrates, which also establishes a gradient and uses the gradient. Does that make sense? Okay, let me know if it doesn't, because this is a fun concept. I like it. Alright, thanks everyone.